Human-Induced Pluripotent Stem Cell Initiative, HipSci, has generated substantial quality control and characterization data on its panel of human-induced pluripotent stem cell lines. This video and its associated document describes the data generated by the HipSci project and how user access to the data is managed. Each cell line generated by HipSci is extensively characterized with genetic, phenotypic, and proteomic data, which is available for use by the wider research community. The HipSci project releases both its quality control and its assay data. The HipSci project runs two quality control assays on the somatic cells and on all the induced pluripotent stem cell lines derived. From this QC, HipSci assesses pluripotency and genetic stability of the lines and selects suitable cell lines for banking. Expression array. The candidate iPSC lines and somatic cells are both assayed using an expression array such as the human HT12V4 expression B chip kit from Illumina. The results of these assays are then used by Pluritest, a tool which compares to expression data from a training set of 450 lines to assess the level of expression of pluripotency markers in the given sample. You can see the Pluritest results for a particular cell line on its cell line page. The two charts show the calculated pluripotency score and the novelty score for the iPSC lines and the somatic cells. Genotyping by array. The second QC assay is again conducted on both the iPSC lines and on the somatic cells. Both are genotyped using a B-chip array from Illumina, such as the human core exome 21V1.1. The results from the iPSC lines are compared with the results from the somatic cells to assess genomic integrity. The comparison is conducted using an algorithm written specifically for the HipSci project. The custom copy number variation results are visible on each cell line page and show any copy number aberrations between the somatic derived control data and the given iPSC lines. The first plot shows a summary of the analysis across each chromosome. The subsequent plots show any aberrant regions in more detail. Based on the QC results, particular iPSC lines are selected to be banked in a cell bank. These lines are then subject to several more genomic, proteomic, and phenotyping assays. These assays are conducted in 3 different institutes from the HipSci consortium. The Wellcome Trust Sanger Institute conducts all of the genomic assays. King's College London generates the cellular phenotyping data, and the University of Dundee conducts all of the proteomics assays. After QC, we undertake 4 further genomic and genetic assays, each yielding different information. The first is whole exome sequencing, which is used to genotype for exonic regions of the genome. This is done in all selected iPSC lines, and it is also done for 250 of the somatic cell lines from which the iPSCs were derived. Whole genome sequencing provides the complete DNA sequence of the genome. Whole genome sequencing data is available for both the somatic cells and their derived iPSC lines from 150 donors. RNA sequencing allows the study of phenomena such as gene expression levels, alternative gene-spliced transcripts, and gene fusion events. RNA sequencing is carried out on all iPSC lines that are selected for banking. Methylation profiling by array is used to probe the methylation pattern of the DNA for epigenetic studies. This assay is carried out on all iPSC lines that are selected for banking. HipSci's cellular phenotyping assays are carried out at King's College London at the Centre for Stem Cells and Regenerative Medicine. Imaging the HipSci cell lines and using high-content analysis, we quantify and standardize phenotypic features that are informative of cell behavior in an artificial microenvironment. Methods have been developed to facilitate the integration of dynamic and endpoint imaging data with genomics and proteomics data. This allows for the study of genetic contribution on cellular phenotype and the association of single nucleotide changes to parameters such as adhesion, morphology, and proliferation. HipSci's proteomics data is generated at the University of Dundee. Mass spectrometry is used to assay the proteome of the HipSci iPS cell lines. We undertake mass spectrometry-based measurements of protein expression in many of the HipSci cell lines from both normal and disease groups. This includes both label-free and TMT measurements of protein abundance. At HipSci, we are committed to making all of our data available to the whole research community. We have an early pre-publication release strategy, which means you will see our data in the public archives very soon after it is generated. Some of our genetic and genomics data have access restrictions based on the donor consent provisions. I am going to explain in more detail about our managed access and open access cell line categories. The lines from our normal healthy donors fall under either managed access or open access based on the ethics conditions they were collected under. All cell lines from donors with rare diseases will always fall under managed access. Managed access data is only available via a data access agreement. For the managed access cell lines, the recipient agrees that any genetic or genomic data it generates from the use of the cell lines will be held securely and only used in biomedical research. The data must only be made available to third-party researchers under a data access agreement at least as stringent as the data access agreements for data from the same cell line submitted to the EGA by Sanger. For open access cell lines, there are no data access restrictions, and the data can be freely downloaded. The recipient of the cell line agrees not to make any attempt to identify the original donors of the cell lines. They also agree to remove any data mapping to the Y chromosome before making genetic or genomic data available to third parties. Once this has been done, data may be made available for biomedical research in an open fashion, permitting redistribution of derived datasets. This is now the end of the video. If you have any questions or queries, please contact us at hipsci@ebi.ac.uk.